So thank you so much for joining us. We were looking forward to seeing you a couple of weeks ago in Melbourne, but this is the next best thing. So thank you for that.
Yeah, I'm very sorry I couldn't come. I was actually looking forward to the trip, but yeah, I fell ill. So
Yeah.
forgive.
I understand maybe later in April next year, perhaps.
Possibly.
Yeah, excellent. So really today we just wanted to get a little bit of some of your insight into the future of the field of bioinformatics. So I teach a unit called Introduction to Bioinformatics to second year biomedical science students. And really we just want to get some insight into where you think the field is going and maybe even a little bit of history and your experience in the field. So I guess my first question might be a little bit about your history with the Human Genome Project and your recollection of that time and what it taught you.
Well, that could be a long story, but I guess in brief, I mean, I'm a classically trained computer scientist.
Yeah.
PhD in 1981, but I've been dabbling in kind of problems involving DNA sequence because it was a kind of very interesting collection of problems about how to deal with long strings and how to think about how to match them. And so really I started out my career as a theoretical computer scientist thinking about, efficient methods for working with string data in general. And that slowly I first became aware of the Genome Project in the mid-'80s. I began to just kind of on a kind of toy level work with those problems. And it basically, I guess it's a trajectory that was pretty common. I got more and more interested in the biology and in the application. By the mid-'90s, I was fairly involved in the DNA sequencing problem and had proposed that we could actually sequence the human genome by shotgunning it, which at the time was thought to be a rather insane thing. Although it really wasn't. I mean, I had done simulation work that showed that if you could basically understand all the repeats in the genome and somehow other factor them out, then basically what I understood was that the rest of the genome would go together quite nicely with the data that we could collect at that time. And so, of course, nobody wanted to do it. But then there was this startup company, Celera Genomics, that was really it was really a ploy by Applied Biosystems to get people to buy their instrument because they were behind the curves. They were behind Amgen. Yeah. So they came up with this idea of sequencing the genome, and they got Craig behind it. And then once I realized they were going to spend $300 million trying to do it the way that I proposed to do it, I didn't feel like I had any choice. So I left academia at that point. I'd been a professor for 18 years at that point. And I went and I think the rest of the story is fairly well known. We were actually quite successful, and now this idea of assembling things is an established paradigm. In fact, it got a little bit crazy because in the 2000s, then, if you go another 10 years forward, we had these short read instruments, these kind of next-gen sequencers that produce data very cheaply, but they produce very short reads. And honestly, they don't assemble well, and so things, I think, completely went to hell. I basically left DNA sequencing, and it was at that time that I got very interested in what could you do with the genomes and how would you interpret them. And well, I know a lot about bioinformatics, traditional bioinformatics, thinking about networks and interactions of proteins and and you can look at DNA sequences. What I realized is that the functioning of cells and of collections of cells is so complex that really our ability to predict anything in silico is quite limited. I mean, it isn't the case that there haven't been successful predictions, but they're fairly limited. Protein A may interact with protein B and control, something X, and you still got to go do an experiment, right? And so, and so what I basically embarked on was, because I saw that nobody else was working on it, was that I started getting involved in trying to interpret imagery from microscopes. Because one of the biggest things that came out of the genome projects, once we, you know, I mean, the young kids may not understand this today, but it wasn't until 2002, that we actually had good sequences of the human, of the mouse, of the fly, of the worm, of kind of all the model organisms. To the extent that one could actually do transgenics on a systematic basis. I mean, before that, you were, you know, you had access to, you know, small pieces of the genome that you happen to know quite well. But now you could basically, on a systematic level, basically go in and basically transgenically modify any protein or instrument it in any way that you can, myriad ways that you can do that. And that's been incredibly powerful, and in fact has become now kind of a major paradigm for a lot of biological researchers. We can basically do what we've always been doing, which is we can perturb the system. We can knock proteins down. We can knock them up. We can watch them, and this has been hugely valuable to kind of giving investigators clues. I mean, there's still, you know, all the biochemical experiments, but back to the kind of the point is what I saw is that it's not just about you know, the chemical interaction network. A lot of what cells do is about physics. It's about what's happening in time and space. It's about the location and organization of these things. So it's the chemical network, the interaction network combined with its spatial locality, and then all of the physics of, you know, adhesion, of force generation, of diffusion, of phase separation. I mean, all of these things are incredibly important. And I realized that I just wasn't like smart enough that I was going to stare at DNA sequence and figure it out. So I basically got involved in trying to interpret, you know, imagery from microscopy, because now you can basically take any genetic entity of interest and you can light it up in the cell and see what it's doing, or at least get some pretty good idea about what it's doing and where it is and when it's acting and who its partners were, simply by observing these things. But what I observed as a bioinformatician is that nobody was looking at this, right? And well, not nobody, but there were a few people, right? So there's the ISMB meeting with 2,000 people all thinking about sequences and graphs, okay? But there's nobody thinking about, you know, imagery, image analysis, computer vision, right? I mean, and it's been a problem because it's a different set of algorithmic tools, and people, you know, they're comfortable thinking about sequences and graphs. They don't want to suddenly have to learn computer vision.
Is it something that's more complex than the genomic work you did? Do you feel it's another level of complexity, or is it on a similar level, your imaging work?
The problems are much harder, actually, because microscopy, I mean, there's DNA data is quite neat, right? I mean, yes, there's some error rate, okay, but it's, you know, it's 5%, maybe 15% with the new long-read instruments, but it's tolerable. Whereas when you're looking at a microscope, I mean, you're limited by the Nyquist limit. You can't see anything smaller than 250 nanometers, and you've got a low signal-to-noise ratio frequently. In other words, there's a lot of noise in the imagery, and so it's very interesting because you have to basically. But we know a lot about what we're looking at oftentimes, and so it's a very interesting kind of subset of computer vision problems in which we have kind of very weak imagery, not very clear imagery, but we know a great deal about what we're looking at. So we have a lot of what would be called prior knowledge. And so, you know, a classic example actually for medicine would be the ultrasound, you know. I mean, the ultrasound of the baby, right? I mean, it always takes me like 15 seconds to figure out where the baby is, right? I know I'm looking for a baby, and you can actually do it, okay? But it's because you know what you're looking for. It's not because that ultrasound's showing you such a beautiful picture of what's in the womb. But anyway, let me try to finish I'm sorry, I started a bunch of threads, so let me try to finish them. So basically, in 2000, I basically drifted away from genomics, from sequencing, and went into thinking about imagery because of for the reasons that I've given. But now recently, I'm actually back into sequencing again because of the long-read sequencers, so the next-gen sequencers. So now there's a collection of sequencers, the Pacific Biosciences machines and the Oxford Nanopore machines, the MinION, that basically are producing very long reads that are very noisy. And what's exciting to me about that is that, again, this idea of building beautiful de novo reconstructions of genomes is back on the table. So that's kind of my story. So I'm now in Dresden, Germany. I'm a Max Planck director. I've gone completely native. I started in a computer science department. I'm now working in an institute surrounded by nothing but biologists as far as the eye can see. And I'm happy as can be.
Happy as Larry. I guess I'll touch on that point about the PacBio and the system. And it was when you heard that it was completely random, the error rate, that you realised you sort of were back in the game, because the perfect long read de novo sequencing was possible in your mind. I wonder if you could talk a little bit more about that.
Yeah. So I mean, basically, back in the mid-90s, when I first proposed whole genome shotgun sequencing, the Sanger reads were about 600 base pairs long. They got to about 800 by the time we did the project, and they were quite clean. They had a 1% to 2% error rate. Now, the error wasn't random, but it was quite low. The average is 1%, 2%, 3%. And if you actually paired them, which was an important very part of the proposal, which is that, in other words, you produced an insert of size, say, 2 kb, and then you sequenced the ends of them. Then you had actually quite powerful information. You had kind of a double-sized read. It was almost as powerful as a 1600 base pair read. And on the basis of those, I did simulation work that showed that everything should go pretty well. Because most of the repeats of the genome are ALU elements, which are 300 base pairs, and the reads are longer than that. So as long as repeats are spanned, things go pretty well. There are other nasties like full-length retrotransposons, L1s in the human, that are about 7 kb, that obviously wouldn't be spanned by single read, but they would be spanned by 10 kb mate pairs, right? These paired reads. And so, I did a bunch of simulations that said, yeah, look, I mean, you're going to get 99% of the thing, okay, organized, right, with this thing. And so we proceeded. So then fast forward to 2002, 2003, 2004, basically the emphasis, we had sequenced the human genome, and now the emphasis was on medicine. And the American programs and the European programs basically decided that the emphasis should be on cheap sequencing.
Yeah.
And so people started to deliver cheap sequencers. The unfortunate thing about these cheap sequencers is that the reads were very short. They were, I won't go into all the technology, but they were initially 30 base pairs long. I mean, it was ridiculous, okay? But they were like, an order of magnitude cheaper, right? And today, of course, it costs, about $1,000 to do a 30x human genome. With these technologies, which are now producing about 100 base pair reads. But that's still too short, okay? They're very clean. The error rate's 0.5%, so they're phenomenally clean.
Yeah.
And you can do a certain amount of work, but you just can't get through the repeats, okay? And so I mean, there's some really, piss poor genomes out there that have been done shotgun sequencing with these things. And I mean, the informatics is quite nice. It's all gone to kind of these de Bruin graphs, which make perfect sense for that scale of problem. But you can't, we were further than ever from the goal of producing high quality reference genomes, right? Which was always the initial goal of the Human Genome Project, right? In other words, I took a huge amount of flack because, they were saying, they were going to produce the Swiss cheese genome, and it's going to be awful. And, and it wasn't actually. It was pretty damn good, okay, because of this pairing thing. And it was a damn sight better than what you can do even today with these new technologies, unless you make a lot of paired end libraries, which are quite difficult for these technologies. And so to me, it was kind of funny that it went from the mindset was is that it can't possibly work, right? In 1996 to, oh hell, it'll work on anything.
What were you thinking?
Okay, in 2005, by 2005 they were, and I was kind of embarrassed. I was like, look guys, this isn't what I was proposing. I mean, I want to produce high quality de novo genomes. And really the point of the NGS period was actually that they wanted to resequence human genomes. I mean, that was really the point. I mean, they shouldn't have called it the $1,000 human genome project. It wasn't a $1,000 de novo human genome. It was a $1,000 resequenced human genome where I'm going to get most of the SNPs. Okay, and I'm not going to actually understand that much more about structural variation between human beings, which is actually turning out to be quite important. Okay, it turns out there's a lot of large-scale variation between our genomes.
Yeah.
And especially like the projects on the cancer. So anyway, I could go on and on about that. But anyway, so in the meantime, there was already early work in around 2002, 2003 on single molecule long read sequencers, both through the pores and then through the polymerase isolated in a zero mode waveguide kind of PacBio approach. And I always kind of wondered what happened to the PacBio idea, but they finally, after 5 or 6 years, basically produced an instrument that was doing fairly well by 2010. And so now we're kind of entering this age where we've got these technologies that are becoming and they're becoming cheaper too, where, we can now produce very, very long reads. They're single molecule reads.
Yeah.
So you're basically observing a single molecule, and they are therefore, again, I won't go into the details, but they are therefore very noisy.
Yeah.
So instead of 0.5% or even 3%, the noise rate on these things is 10 to 15% or worse. Okay.
Which would have put you on off.
Beg your pardon?
Which would have put the commercial viability off. That an error rate like that. A lot of people thought that was unviable.
Yeah, and so and that was a mistake actually, because I mean, so people thought, because if you have a 10,000 base pair read, and you give me a 15% error rate, the question is like, when is a match significant? So, I have two pieces of sequence that are kind of error. If I have, like, for example, a 2 or 3% error rate, then if I have like 40 to 50 base pairs that align at 5% error, that's statistically very significant. That's very unlikely to be an accident. And in fact, it's the case that it's probably either because those two reads came from the same place of the genome, or because they came from two similar copies of a repeat. But statistically, that overlap, that 40 base pair overlap, that's a real signal. Something's going on there. It's either they belong together because they came from the same part of the genome, or that's a repetitive element and they both came from the same part of that, similar parts of that repetitive element, right? It's not because it was an accident. It was just that chance thing that, these sequences happen to align. Well, okay, go to 15%. Well, how long does it need to be? Well, it needs to be 1,000 base pairs. All right. So now you need, but at 1,000 base pairs at 70%, okay, it's statistically just as significant as that 40 base pairs. Those two reads belong together, no doubt about it. Okay. So basically, and now I've got a 10,000 base pair read. So I'm going to have lots of 1,000 base pair overlap. So basically, there's no problem understanding how these reads go together. And then if you can understand how they go together, then the question is, like, if you have them piled deep, if you do 50x sequencing and they're piled 50 deep, then you have 50 votes for what the base is at every place. And there's a long-standing theory from Waterman and Churchill from 1988 that basically says that if the error is random, in other words, if the individual places, whether it's an insertion, deletion, or substitution, are random, then the error rate of the consensus decreases geometrically with the depth. So in other words, by the time you're 50 deep, The odds are, 1 in 100,000 that it's a mistake, that your consensus is a mistake if they're random. Now, and here's the thing, is that actually every technology up until these technologies was not making random error. There were places, like there was for Sanger and for even, Illumina, there's a location every 10,000 base pairs or so in an underlying genome where the sequencer is going to make exactly the same mistake over and over and over again. Well, so now if you vote, you're going to get that mistake.
Yeah.
Right. So you can see the difference. So there has to be a predisposition to a particular error at a particular location. But that's true of the other technologies. It is not true of the PacBio, okay, which means that it's random. So in fact, what it says is if you're willing to sequence enough and go deep enough, you can be arbitrarily accurate. Okay? And so I think that kind of it was naive to assume that high error rate meant bad data. Okay? It does not mean bad data. It just means more difficult computational problem. So which to me is great. I'm a computer scientist.
The problem that needs to be solved.
I got an interesting problem, right? Nobody's built an assembler that works at 15% error rate. I'm bad, and I'm excited about this data because because of the length, right? Because most of the repeats are spanned, means that I'm going to be able to produce near perfect reconstructions of genomes. And that's been borne out. I mean, basically they did 100x of Drosophila and out of the box with the current assemblers, which are still being improved because we still haven't built the best possible one for this data. We were getting basically a result that was equal to release 6 of the Drosophila reference genome, which is the best genome, okay, currently in existence on the planet, okay, out of the box. So what this means then is that the novo genomic sequencing is back on the table, okay, and it's currently a bit expensive. A 50x human 3 years ago would have cost you about 50 grand. Today it costs you about 10. And by 2018, it will cost 2. So it's a kind of the usual thing for these technologies. They get cheaper and cheaper, and better and better. You don't have to be a futurist to know that that things get cheaper and faster with time.
Okay, so on that point then, so I remember back in the late 90s, I was only quite young, but in the early 2000s, there was a real excitement about this technology and genomics and the potential and also the fears of it. And now here we are 15 years later, and a lot of those possibilities have arrived. We have, the cost is down, the speed is up, the quality is getting better and better. We've got technology like CRISPR to edit the genome, but yet I don't see the same excitement from the public or even from some areas of biological sciences for genomics. I wonder if you have any ideas of how how that's changed over time, in your opinion.
Well, I'm not sure about it. I mean, I personally think that bioinformatics is fascinating and it's got quite a future. And I think that it's rare in life that there's a clearly stated goal that captures the imagination of the public, and sequencing our genome was clearly such a statement. It's very nice, I'm going to sequence the human genome, right? We are going to sequence the human genome. Right? Everybody can get it. I mean, what would be the analogous statement today? I mean, that's kind of the issue, right? I mean, it's not that there isn't fascinating work going on and that there aren't beautiful questions, but I mean, I guess you could make statements like, I'm going to be the Linné of my time and sequence the entire world. I'm going to see, so they're like the G10K, B10K projects, but they don't, it doesn't have quite the cachet, right? I mean, hey, look, I mean, you give me a good one-liner and I'll go after it. Believe me, I spend some time thinking about it. Although mostly I'm having a great time, building microscopes, interpreting images, and then working on this kind of next gen sequencing. I mean, that's my agenda, and I'm having fun. And I think the field is, I think it's getting very interesting. The stuff with the organoids is fascinating. This idea that we can take stem cells and basically let them go, and they produce kind of things that sort of look like brains or stomachs or kidneys. I mean, this is fascinating, right? And so I think kind of in the future, our ability to manipulate tissues, so tissue engineering, is going to be incredible, right? And I think we're going to learn a lot about that. I think there are beautiful questions still that remain in development. I mean, we still don't understand how the genome coordinates the activities of hundreds of thousands of cells to produce with incredible fidelity a given shape and form, right? I mean, this is an outstanding question that I think is fascinating. But yeah, I mean, finding a crisp statement, and it's also the case that I think there's been a lot of kind of inflation. I really there's been a tendency for kind of one-upsmanship, and I think a certain amount of overstatement in science about where you're going. I mean, everything has to be, I think I'm a little exhausted by how superlative and top class and how big every project has to be. I mean, I'm just a guy that wants to figure out a few simple things, and so especially in the U.S., I think there's a tendency to overstatement, and it's getting a bit cliched, and that may be part of it. We're just all kind of ennui, and I mean, I think in general our society has a lot of problems beyond science. I mean, we're in I'm not quite sure what's happening in Australia, but I know that in the States, the average man is having a pretty hard go of it. And, there's a lot of corruption. Our systems don't seem to be working. I think we're very near a financial collapse or some kind of cataclysmic monetary event, given the policies of the world. I think we're entering. I think we're entering a different phase of mankind, and so for the young people, I mean, good luck, and I hope you do better than us.
That's a lovely way to put it.
Sorry, that was a little bit philosophical, probably a little bit, but for early in the morning. But that's my opinion. I'm sticking with it.
No, that's fine. One last question, I guess, about I can't talk to you and not ask you about it. This is something that our students and I've seen the citation index for your the paper in 1990, and it just keeps going up and up. Is that indicative of the field of bioinformatics being more and more relevant to biology, do you think? And did you ever see it becoming such a success?
Well, I think bioinformatics is it's critical to biology. I mean, I think that biology is going to become ever more quantitative and analytical, and therefore digital. Okay, because we are intrinsically talking about systems that are so complex that the only way that we're going to be able to kind of do predictive things around them are to simulate them, right? I mean, and frankly, even the simulations, or even if we knew everything we needed to know to do an accurate simulation, the computer power still isn't there, but it will be. Okay, I mean, I have colleagues who are doing very interesting simulations. I mean, I would encourage people to who are entering the area to be a little bit careful not to be basically just a discrete mathematician. Okay, I would, or to just understand sequences. I would, especially if you're still kind of in your early stages of training, I would make sure you understand some physics.
Yes.
I would really make sure you understand some physics, and you understand how to model, because I think those things are going to be very important going forward. I think that also you need to be the people with the juice are going to be the people that understand physics, that understand chemistry, and can understand these things from first principles. I think that we do a lot of biology has kind of become what I call kit biology, right? I mean, you run protocols that have been well worked out, and you buy the kit, you run the protocol, and so you learn how to run the protocol. But the advantage in the biosciences has always been to the groups that have the best methods for interrogating the systems to answer the question. It's always been about having the tech, okay, or methods to get into. So if you're running a lab and your repertoire of things that you can do is exactly the same as the repertoire of the things that the other, 10,000 biologists in the world can do, then what's your advantage? Okay. So, think about that. So is your advantage could be your question or your system, right? And so there's a certain, picking the right system, picking a good question that will yield to the techniques that you have available to you is important. But having techniques, okay, that are unique to you or to a small group, okay, or to be developing techniques. So I would encourage future biologists to think about educating themselves in a way that will allow them to not only understand the domain, in other words, what's true about biology, but to understand the repertoire of available techniques and technologies behind that. So to have something special in your back pocket, to either know something about microscopy or optics, to know something about physics and physical modeling, because I think that's going to be the biology of the future. So I guess that was aimed more at biologists. There are the bioinformaticians.
Yeah.
Bioinformaticians, you've got to, you've got to get away from strings and graphs. Okay. You've got to understand, you've got to know some image analysis and computer vision. You better know some AI, some machine learning and artificial techniques. Deep nets are all the rage, a little bit too much the rage. Again, things tend to be a little bit too trendy, but deep nets are important. So, having these repertoires of skill, and then, my most general advice is simply just train yourself so that you're in a position to learn whatever it is that you need to learn. All right. The real point of an education isn't to come away with a fixed repertoire of things that you know about, okay, and that you understand, but to really understand how to learn and deeply think analytically about what's happening. It's about the process. What you're really doing is you're refining your ability and your process to learn and to acquire any knowledge that you need to acquire. And I really think that's the meta level. Sorry, I'm getting to be an old man.
Oh, no, that's fantastic.
Philosophical, but I hope that was...
that's brilliant. All right, thank you so much, Miles. I know you have to get to Berlin, and thank you for giving us the morning.
Yeah, it's fun. Okay.
Great.
All right.
We look forward to seeing you in Australia sometime soon.
Yeah. Sorry I didn't make it.
No worries at all. Thank you so much.
Take care, guys.